Hello everyone, my name is Eric Willems and I'm a staff scientist in our cell biology R&D group at Thermo Fisher Scientific. Today I'll share some of the improvements that we've made in building iPSC-derived disease models. Everyone knows that ever since Yamanaka came up with the reprogramming of somatic cells, that disease models have taken off in the field and now allow us to generate any kind of differentiated cell type from a diseased patient through reprogramming. After having the differentiated cell types, we can then look for disease phenotypes in a dish and then ultimately put those cells in a screening assay to discover drugs for treating the patient's disease. So really, building a cell model is very useful to predict inputs and outputs from a particular disease, and hopefully in the long term, these sort of assays can then be used to translate treatment in organisms such as humans. So Thermo Fisher, over the years, has put together a wide portfolio of tools, workflows, and services to address the iPSC disease modeling field. So we have a lot of tools in terms of characterizing patient samples, such as our next generation sequencing equipment. We have our reprogramming kits, such as Cytotune, where you can now take a somatic cell from a patient and reprogram those into pluripotent stem cells. You can then culture those pluripotent stem cells in a variety of media that we have, as well as a number of matrices. We have reagents and analysis tools to characterize your stem cells. We then have genome editing tools so you can engineer the genome of those cells. And furthermore, we have differentiation reagents where you can then start turning your pluripotent stem cells into your cell type of interest, which can then be used to run biochemical and cellular assays using one of our instruments or reagents. So we really have a wide variety of tools that can be used in a pluripotent stem cell workflow. And furthermore, the majority of the tools that we have are also being implemented in services that we run internally for any customer, academic, biotech, pharma. So today my presentation is going to focus on the new innovations we have around genome editing in pluripotent stem cells to generate isogenic disease models. So typically when you're talking about isogenic disease models, you're always thinking about comparing a healthy donor with a patient donor. However, there's a high variability between different patients. So with genome editing, we can actually get around that now. So for example, we could take a healthy donor, derive its iPSCs, and we could then use genome editing to introduce particular mutations to then mimic a diseased donor sample. And in the opposite direction, we could take a patient sample, derive the iPSCs again, and then do a genome edit to correct those disease-relevant mutations so that you now have a matched isogenic pair coming from the same donor. So about 5 years ago at Thermo Fisher, we started a collaboration with the Parkinson's Institute to try and do exactly that. So we had a number of patient fibroblasts that were carrying mutations relevant to Parkinson's disease. And we then wanted to try and revert some of these mutations using genome editing tools that were available at that time, and then further differentiate those cells into neurons and look at some phenotypic differences between the disease sample and the genome edited control. So we took those patient fibroblasts, we used our Cytotune reprogramming kit to turn those fibroblasts into induced pluripotent stem cells. We then grew those pluripotent stem cells using our Essential 8 medium and vitronectin matrix, which were top of the line at that time. And we then used our TAL-based genome editing system to deliver SNP corrections into the genome of these cells. We then further differentiated them using our neural stem cells and did some relevant assays. And while in the long run we were successful in doing this, it took us about 1.5 years to go from a patient fibroblast to a successfully edited pluripotent stem cell. So we identified some clear bottlenecks, including the design of the genome editing tools themselves. So the talents that we had back then were DNA-based, and also for introducing the SNPs, we required a larger donor. And using those two DNA-based methods typically yielded very low editing efficiency, both at the level of indel formation as well as homology-driven repair. And then secondly, for DNA delivery, there are several caveats in the pluripotent stem cell field as well. First of all, lipid-based methods are not very efficient. Electroporation was the only option, but electroporation is somewhat stressful to stem cells. And then furthermore, delivery of DNA causes adverse effects such as spontaneous differentiation or even genomic instability in some cases. So the tools and the delivery was a big problem and was not setting us up for success downstream in terms of isolating clones, but the isolation of clones itself is a challenge as well. So back then, media systems did not really support single-cell sorting, so we were forced to do manual approaches where we would manually isolate clones by cutting them into pieces and expanding one piece further in culture and using the other piece for genomic DNA analysis. But then, because of that manual picking, clones were often found to be mixed populations, and due to the fact that these populations were mixed, TACMAN, qPCR assays, or Sanger sequencing assays were always difficult to interpret because there were those different cell types in that clone. So really, the pain in the whole genome editing process was, on the one hand, was the delivery of the tools and the tool itself, but at the same time, the clonal isolation and screening had an equal problem in the whole workflow, which made it a laborious and iterative process where you had to go back several times to get to a success. So in the past couple of years, Thermo Fisher has worked on a number of innovations to improve that genome editing to help in the iPSC disease model construction. So at the level of genome editing tools, we developed a Cas9 protein called TrueCut Cas9, which works very well for genome editing in pluripotent stem cells. In addition, we released perfect match TALs, which are an RNA-based TAL method, which is easier to deliver than DNA into the iPSCs. And then third, we also have a Cas9 iPSC line, which is a iPSC line that has a stably expressing Cas9 protein. And I'll come back to these different tools in a minute. And then we also have delivery tools. We have our Neon electroporator, and then we also have a lipofection-based system, lipofectamine stem, that would help in delivering the various editing tools. And then for clonal isolation, one of our biggest innovations there is our StemFlex medium, which is a medium that was designed to promote single-cell survival of iPSCs, and that has now allowed us to do single-cell sorting using FACS of iPSCs to reliably isolate single-cell clones. And then third, as I mentioned previously, we have a number of kits and reagents that allow you to look at your disease model and analyze possible phenotypes. So we have differentiation kits for cardiomyocytes, dopaminergic neurons, and other CNS neurons. And then we have a large variety of assays, including cell health assays, other cell-based assays, and we also have high-content imaging instruments, and so on. So right now, I'll just deep dive a little bit in some of these technologies and kind of show you what we achieved with those new tools. So the first new tool is our stable Cas9 iPSC line. So what we did is we took that iPSC line and tried to introduce a number of SNPs, as well as just doing indel introduction by just introducing the guide. And so what we used on the one hand was a genomic cleavage assay to just look at the indel formation, and an example of genomic cleavage assays are shown on the top in that DNA gel. And on the other hand, we looked at SNP introduction efficiency using targeted next generation sequencing. And on the right-hand side of the slide, you have an example of what a next generation sequencing result looks like. So you can clearly see between a wild type and a sample that has some SNPs that we can clearly detect the presence of SNPs on a number of alleles. So when you look at that from a broader picture over 9 targets that we looked at here, for all of those targets, we typically get about 20 to 80% indel introduction efficiency. And as you can appreciate, that number is highly target dependent. And furthermore, when we look at SNPs, we typically get somewhere between 5 and 40% homology-driven repair. So the Cas9 iPSC line is really an efficient tool to introduce a high number of mutations using one donor background. However, we understand that sometimes you want to do editing in your own iPSC or in a specific patient-derived iPSC. So the tools that we derived there was our TrueCut Cas9 protein. So it's a protein that then complements with a guide RNA, and that guide RNA protein complex can then be delivered to the cells. So on the left-hand side, you see the efficiency of SNP introduction on 3 different targets, and using 3 of the tools that we've had in our catalog. And you can see that the stable Cas9, as I've shown in the previous slide, performs really well. And then our first generation Cas9 protein did not do so well in pluripotent stem cells, but then our second generation TrueCut Cas9 protein works very well and matches the efficiency of the Cas9 stable line. So genome editing in your own iPSC line now becomes much easier. And in the left-hand side, everything was delivered using our neon electroporator, but we understand that the neon electroporator is not always available to you. So we then also developed a lipofection-based reagent called lipofectamine stem. And when we compare these 5 different conditions here, looking at different genomic targets in 2 different iPSC lines, what you can generally appreciate is that the neon is really the better system to achieve efficient genome editing, but at the same time, lipofectamine stem allows you for 3 of those 5 targets, you can still get fairly efficient genome editing to the extent that you can still successfully isolate clones. Which then brings me to the innovations that we've done around the single-cell clonal isolation of pluripotent stem cells after doing the genome editing. So you always want to isolate a single-cell clone after the genome editing, just so that you can have homogeneous cell lines, either being a homozygous edit or a heterozygous edit. So we spent quite a bit of time in trying to optimize single-cell sorting on pluripotent stem cells to get to reliable single-cell isolation. Because in the field of 293 cells or cancer cell lines, using FACS for single-cell isolation is very common. So we really try to achieve the same with pluripotent stem cells. So we first set up a very stringent gating strategy where we looked at different forward and side scatter gates to remove debris and doublets. And then we furthermore introduced a double gating strategy using propydium iodide and Tra-160 antibodies to be able to reliably sort viable pluripotent stem cells. And after we had set that up, we then looked into different cell substrates, different cell media, and different supplements to try and see whether any of the post-sort conditions could help in promoting survival of pluripotent stem cells after the cells going through a cell sorter, which is typically a stressful event. And in a nutshell, what we observed was that under standard conditions, and now I'm looking at the histogram plot on the left, that the standard media conditions such as essential 8 and Jaltrex didn't really reliably yield any single cell clones. However, when we started adding our Revitas-Cell supplement as well as laminin 521, we noticed that we could slightly increase the efficiency to 10 to 15% of single cell survival. However, when we introduced our new StemFlex medium into the system, we were all of a sudden getting clone survival in the 30 to 50% range, which is really a dramatic increase in the efficiency. And as you can see on the images in the right-hand side, the single cell clones have a normal pluripotent morphology and are staining really nicely for pluripotent markers. So really, by using the FACS system, we were able to bring down the clonal isolation from a several-month process with having to isolate manual clones several times to get to your clonal line. We now brought that down into a process that takes about 2 weeks, which is a dramatic improvement. So we then used this single-cell sorting method on some of the disease SNP pools that we had made in the stable Cas9 line, and we isolated somewhere between 200 and 300 clones, and then screened those clones by sequencing to understand what the distribution was of the different heterozygous, homozygous, unedited, or random indel clones in that selection of pools. So as you can appreciate from the 5 targets shown here, when we do the editing and do the clonal isolation, you will typically, with different degrees of success, you will typically identify a homozygous SNP using the editing methods I had described. However, you'll appreciate that heterozygous SNPs, shown in blue, are not always found. And then in all pools, evidently, we are detecting indels still where the homology-driven repair did not occur. And then there's also a number of unedited cells in those clones. And there is some correlation between the overall editing efficiency at a particular target and the total success of indels and or homology-driven repair. So then with this, I'm going to sort of switch to one of these disease models and kind of present you a little case study that we've done in the development of a model for Parkinson's disease. So what we did is we introduced the LRRK2 G2019S mutation using the tools I had described. In our Cas9 iPSC line, we then used our StemFlex medium with laminin 521 and Revitas-Cell to isolate the single cell clones. And we then took those edited cells and put them through our differentiation kit for dopaminergic neurons, and then we ran some assays to see whether we could reveal any functional phenotypes that are known to be associated with the LRRK2 mutation. So before I go into the actual differentiation and assays, just briefly want to highlight the importance of characterizing your edited stem cells after doing the genome editing, because both the genome editing and the single cell sorting are stressful events to the cells. And so we did a number of characterization assays to make sure that the pluripotent stem cell lines that we had derived carrying the mutation were normal iPSCs. So the first assay that we looked at is our TACMAN PCR-based scorecard assay. So it basically assesses the pluripotency of the cells. And as you can see in between the red and the black dots, there is virtually no difference between the two, showing that the edited iPSC is not different from an unedited iPSC at the level of pluripotency. And then this was Also further confirmed in our high-resolution pluritest assay, which is a whole genome microarray-based platform that assesses pluripotency. We then evidently also looked at the sequence, both at the level of Sanger sequencing to make sure that the edit in our final pool was still there. And at the same time, we also looked at the sequence using next-generation sequencing so that we could look at 10,000 products of the different alleles represented in the cell line to make sure that the line was clonal. And then lastly, we looked at our karyoStat assay, which is a digital karyotyping assay, to make sure that there was no genomic instability that occurred during the editing and or clonal isolation via FACS process. But in general, what we observed is that pluripotency and the karyotype were unaffected by the genome editing or single-cell sorting, and we found that the genome edit was maintained in the genome and that the line was indeed clonal after one run of single-cell sorting. So now that we knew that, we then moved on to differentiating these iPSCs into dopaminergic neurons. And here is some example immunostaining of cultures containing dopaminergic neurons, both for the Cas9 wild-type iPSC line as well as the Cas9 iPSC-based mutant on the top. And what you can see is there's clear expression of TH, which is a dopaminergic specific marker, as well as beta-tubulin, which is a more general neuronal marker. So we then took those neurons and replated them and then tracked the outgrowth of neurons over time. So the graph on the left-hand side is basically showing you the neurite length per cell over time. And what you can see in the early outgrowth of the neurons, which is indicated by the red circle and line, and directs you to the top graph, you can see that the LRRK2 G2019S outgrowth of neurites is delayed compared to a Cas9 control and even some of the other dopaminergic disease mutants that we've looked at. And if you now wait until neurites have outgrown fully and sort of stabilized in terms of length, we then were even able to distinguish some different phenotypes where we found that the LRRK2 mutant had overall longer neurites, whereas the SNCA mutant that we've also worked at had shorter neurites. We then also looked at the sensitivity to 6-hydroxydopa, which is a very well-established phenotype for dopaminergic neurons related to Parkinson's disease. And using a apoptosis assay, which is our cell event caspase 3/7 assay, we then try to understand whether this is indeed the case with our disease model. So the way the assay works is that in a normal condition, you would have no caspase exposed or not much caspase exposed. As you can see, under DMSO condition, the majority of the cells are blue. However, when you introduce a cytotoxic reagent like staurosporin, you get all the caspase exposed, which can then be detected, resulting in a green signal. So you clearly see between the DMSO and stauro, you see a clear increase in the green signal. And similarly, with hydroxydopa, we see the same effect. When you now quantify that in a normal media condition where glucose is present, you can clearly see that the number of apoptotic cells is highly increased in the LRRK2 G2019S mutation. However, when you now stress the cells a little bit more, by removing the glucose, you see that the LRRK2 G2019S mutant still has high levels of apoptosis compared to control, but now the other SNCA Parkinson's mutant shows the same phenotype. So there's a clear correlation between 6-hydroxydopa toxicity and the presence of Parkinson's-related mutations. We furthermore looked into some gene expression profiling, where we looked at genes that are uniquely associated with LRRK2 mutants. So in the plot on the left-hand side, you see a panel of genes that are known to be upregulated in patients with LRRK2 G2019S mutations. And as you can see across the board, they are overexpressed in our system, compare and control with the LRRK2 mutant. And what's even more interesting is that these genes are not upregulated in our SNCA mutants. So this clearly indicates that this gene expression that we're observing in the LRRK2 mutant is specifically related to the LRRK2 mutant. So with that, I'm going to end and summarize what I've covered. So as I've shown you, we have a number of new genome editing tools, clonal isolation tools, and disease model generation tools that allow you to reliably generate a disease model and then study the phenotypes in an in vitro setting. And with those new tools for genome editing, we now support higher cleavage efficiency and homology-driven repair, and these tools also generate less stress on the cells, and so the spontaneous differentiation and genomic stability is not affected as much. We then have new tools for the clonal isolation, where you can now singularize and single-cell sort pluripotent stem cells. So clonal isolation is no longer a bottleneck in the genome editing workflow. And heterogeneous populations are rarely found, since FACS sorting is a highly reliable method, and one round is sufficient to isolate fully clonal lines. And then I've shown you that using our differentiation kits, you can then show that these genome edit generated models can be differentiated into a relevant cell type, and you can then run a number of assays. And really what these tools now allow you to do is to really make a disease model much faster with less hands-on time. So we're talking about going from a process that took about 1.5 years with a lot of hands-on time to a process that now takes 4 to 6 months with a lot less hands-on time. So with that, I would like to thank you for your attention, and if you have any questions, please submit the questions, and we will respond to those questions by email. Thank you.